With respect to the nervous system, so far we've spent an awful lot of time on the physiology and the microscopic structures. And it's kind of been in reverse of how we tackle most systems. Most systems will do the anatomy, then we'll move into the physiology. But the nature of the nervous system just leads us to do it in the reverse order. So with most of the physiology behind us now for the nervous system, we can move into more gross overall structures and arrangement of the nervous system as a whole. And we're going to start doing that by looking peripherally and moving centrally. So in this lecture, we'll look at the structure of peripheral nerves. We'll move our way centrally and look at the structure of the spinal cord. And once we have the nerves and the spinal cord under control, we can look at how those play a part anatomically in providing the pathway for our reflexes. Then we're going to move up the spinal cord into the brainstem and the brain. However, brainstem and brain will be a different topic. If you see on the screen here now, we see this is nervous system part 1. Brainstem and brain will come in nervous system part 2. So here we'll be looking at the structure of the spinal nerve, the function and structure of the spinal cord, as well as the reflexes. And while we're on the spinal cord, we'll start to look at membranous structures that surround the central nervous system called the meninges. Now, we've stated before that the nervous system is an excitable system, and it's one of our controlling systems. Very early on in this class, we looked at our homeostatic feedback pathway: receptor, control center, effector. And I emphasized before just how much of the nervous system has put its fingerprint on that entire feedback loop, because our receptor is going to be nervous tissue. The communication between the receptor and the control center is going to be nervous tissue. The control centers are generally nervous tissue. It's not until we get to the communication between the control centers and the effector organs where we move away from the nervous system. That communication can still be nervous, but it can also be endocrine, that you'll get to in A&P2. And then, of course, the effector organ itself is essentially going to be anything but nervous tissue at that point. But we will use our nervous system as a means of detecting changes internally and externally, and bringing that information into our central control centers and giving us an opportunity to respond to those changes so we can maintain homeostasis of the body. We'll look at a couple of different ways we can divide the nervous system up. First is going to be structural. We can have a central nervous system, brain, spinal cord, anything that's medial basically, and peripheral, nerves that extend beyond the spinal cord. And if you remember back to some of your axial skeleton anatomy, you would have looked at the skull and the vertebral column. Any nervous tissue found within those structures are going to be considered central. As soon as those nerves pass through an opening in the skull or pass through those intervertebral foramen on the lateral sides of the spinal column, the vertebral column, then we move into our peripheral nervous system. We can also divide it up functionally. Sensory versus motor. Sensory bringing information about the environment into our central nervous system. Motor being the output. And you'll see us refer to a couple of terms called afferent and efferent. Afferent will always be a word meaning to bring into. Motor will be efferent, to move away from. We'll even use those terms sometimes in a circulatory manner. A blood vessel carrying blood to an organ is going to be the afferent blood vessel. The vein draining blood from that organ is going to be the efferent vessel with respect to that organ. But input and output. Now, once we look at sensory versus motor, we can further divide it up again. We will look at, first, our somatic half of our nervous system. That's going to be the nervous system responsible for sensations that we perceive: deep touch, light touch, pain, vibration, the special senses like vision, hearing, taste. It's also going to be related to the somatic motor, the voluntary nervous system. That's the motor system by which you voluntarily control skeletal muscle. The other half, the non-somatic, we are now going to call the autonomic, the involuntary nervous system. Now, you may have heard that referred to already when we hear things like fight or flight. From a sensory standpoint, the autonomic is going to be associated with the visceral organs. It's going to be sensing things like blood gases, blood glucose, blood pressure, things that have to do with internal regulation. Autonomic motor, the involuntary motor, will go to glands. It'll go to the heart to change the heart rate. It'll go to smooth muscle of the blood vessels to get them to constrict and dilate. It'll go to the smooth muscle of the bronchioles in the lungs to get them to constrict and dilate. So essentially, we're looking at it structurally: skin, muscle, musculoskeletal in the somatic region versus the visceral and organ in the autonomics, but also a voluntary versus involuntary description. Now, of course, we've already looked at the nerve cells, right? And there's really no need to beat a dead horse on those. But I do want to remind us about the structure of the neuron. Remember, we had all of those long projecting axons, dendrites projecting away from the cell body. Well, when we look at a nerve is essentially a bundle of axons bound together in a single anatomical sheath. Now, depending on if we're talking about a sensory neuron bringing information into the central nervous system, or a motor neuron carrying information out, will depend on where the cell body of that neuron sits. And it's hard to describe those right now because we haven't talked about the spinal cord. But we'll refer back to this again in a little bit. But if we're talking about a motor neuron, the cell body for that neuron will always be in what we call the anterior gray horn of the spinal cord. It sits directly in the spinal cord itself. The sensory neurons have their cell bodies just outside the spinal cord in what we call the dorsal root ganglion. And when we do a cross section here now, you are going to see some terms that are going to sound familiar. We want to make sure we don't confuse the terms. If you look real quick, it looks like we're rehashing some muscular terms. And we can put a lot of parallels between the structure of the cross-section of a nerve and the structure of the cross-section of a muscle. Because when we cut this nerve, we see that the nerve is divided up into fascicles. Direct rehash of a term there. When we cut across a muscle, we saw the muscle was compartmentalized into groups of muscle cells called fascicles. Here, the nerve is bundled together into groups of axons called fascicles. In the muscle, we had an epimysium surrounding the entire outside of the muscle. Here, we have an epineurium surrounding the outside of a nerve. In a muscle, we had perimysium surrounding each fascicle. Here, we have perineurium surrounding each fascicle. And within the fascicles of a muscle, in between the individual muscle cells, we had endomysium. Here, we have endoneurium. Now, of course, we're not dealing with muscle tissue. So even though we've got a lot of parallels here with the naming, the functions are obviously going to be different. Epineurium and perineurium are protective against physical distortion. We do not want a lot of compression and distortion applied to the axons within these fascicles. The epineurium and the perineurium protect against this distortion. We got a nice, relatively thick epineurium on the outside preventing compression. The perineurium surrounding each individual fascicle will also protect against compression. It's a little less clear as to exactly how it does that, but we know through research that it certainly is the case, because we could do cross sections of some nerves that are more monofascicular. When we do their cross section, there are fewer individual fascicles compared to other nerves that are multifascicular. And we know those multifascicular nerves can handle compression so much better than the monofascicular ones. As a matter of fact, we tend to see a bit of a correlation between the more protected a nerve happens to be, the fewer fascicles we actually observe. Again, implying that if it's protected against from other structures like bone and such, there's less of a need for that fascicular structure. So epineurium, perineurium are there for the physical protection, resisting distortion, resisting things like stretch of a nerve, anything that would physically distort the axons inside and give us unwanted mechanical depolarization. Now, the endoneurium takes on a function very similar to the endomysium that we saw in muscle. Muscle cells were an excitable tissue. Neurons are excitable tissue. We use this endoneurium, again, to electrically insulate one axon from its neighbor. These nerves are peripheral. And again, our theme here is we're going to move from the peripheral into the spinal cord and then start to ascend. All of the nerves we see peripherally will start to converge as they approach the spinal cord. And you will see some distinct convergence that we call plexus, where we have all these individual nerves that will innervate a muscle or supply sensation to a particular part of the skin. And we trail those up towards the spinal cord, and you will see they all converge into nerve roots. And I guess converge, diverge, I guess it all depends on perspective. If you're starting at the spinal cord and working your way out, we're diverging. If you're coming back in, we're diverging. Going out, you're converging. As we branch and then unbranch again. Each of those nerve roots enters the spinal cord, and from there, inside the spinal cord, you'll see we have some axons now ascending the cord, bringing information up to the higher parts of the brain, while other parts of the spinal cord are going to be carrying axons that are descending and looking to hop in a peripheral nerve and head its way out to a peripheral organ. We use these plexes as a way of getting parallel innervation to an organ. Keeps us from putting all of our eggs in one basket. If we look at something like let's look at the musculocutaneous nerve, innervates the bicep muscle. If we trail from here back up to the spinal cord, you will see that we can take various paths and wind up at the C5, C6, or C7 cord level. So as a motor nerve, axons will leave all three of those regions and go through the various converges and diverges in the plexes. And as an end result, if we did a cross section of the musculocutaneous nerve, we would see neurons from each of those three spinal nerve regions. So if something happens, traumatized, and you lose one, you still have two unaffected origination points to provide innervation to our bicep muscle. This is a good time to bring up that in lecture, I will not make you responsible for any individual nerve names. That is a topic that you're going to be covering in lab. They will pick out which nerves and their names you're going to be responsible for. Just like we did with muscle, we're going to keep any naming very generic and look at the overall structure rather than the gross anatomical names. So our nerve root approaches the spinal cord. Now we can start to see where the cell bodies were located that we mentioned earlier. As we approach our spinal cord, you will see that spinal nerve splits. We have a posterior rootlet for sensation. So any sensory neurons that were out here in our peripheral nerve, as they are going towards the spinal cord, they take this posterior branch into the spinal cord. Here is our dorsal root ganglion. That's where we find the cell bodies for any sensory neurons. So sensation will always go through our spinal nerve and enter the rear of the spinal cord. Meanwhile, here is our anterior gray horn. You can see our spinal cord is divided up into white matter and gray matter. We'll get to that in just a moment. But where I put the X is the anterior gray horn. That's where the cell bodies for motor neurons are going to exist. They will send their axons out the anterior portion through the anterior rootlet, where then it will join up with the spinal nerve. So again, our spinal nerve can have sensory and motor neurons running in the same nerve sheath. But when we get through our intervertebral foramen, we split into a posterior rootlet where sensory neurons will enter, and an anterior rootlet. That's where the motor neurons will exit the spinal cord and join our nerve root. Carrying on with more of our spinal cord anatomy. Gray matter, white matter. White matter in the spinal cord will be superficial. The gray matter is this H or butterfly shape, however you want to describe what it looks like. The difference here is going to be the presence of oligodendrocytes. Remember, those were the same as Schwann cells. They wrap themselves around the axons. So just by knowing that alone, you should guess that anywhere where we see white matter, those are going to be myelinated axons that, depending on if they're sensory or motor, are either ascending the cord going towards the brain or descending the cord coming from it. So anywhere where we see white matter, those are myelinated axons. And if we look at the white matter, I just jumped ahead to a diagram, you'll see that the white matter can be regionally broken up, mapped, if you will. We can have our spinothalamic tract as a sensory tract containing neurons for light touch, pain, moving up the cord towards the brain. We have our corticospinal tracts carrying motor axons coming from the brain, going down the cord, that'll branch out the cord and go out to the muscles. But anywhere where you see white matter are axons ascending and descending the cord. Now, by default, if you see gray matter, you're looking at an area that doesn't have myelin. And visually, that should make sense. We know that that myelin is a fatty substance. It's going to have a white, more glistening looking appearance to it. You remove the myelin, and you're left with this dull gray structure. So anywhere where we see gray matter, we're going to have cell bodies, like our motor neuron cell bodies that are going to exist out here. We're going to have dendrites, or we may even have unmyelinated axons. Because remember, not all axons contain these myelin sheaths. Now, regionally, we divide our white matter up into funiculi. We had a posterior funiculus, we have a lateral funiculus, we have an anterior funiculus. Now, I know we're dealing with the spinal cord here, but I guess we can still use the pun that we're not dealing with brain surgery here when we realize that those funiculi are just named based on their directional orientation. Likewise, we look at the gray matter, and we have a posterior horn, we have an anterior gray horn. If we were dealing with a part of the spinal cord that was more so in the thoracic region, we would even have a lateral gray horn here. We'll get to that once we do the autonomic nervous system. Connecting both halves, we have a gray commissure. Commissure meaning connection. And right in the middle of that commissure, see that little black dot? We have our central canal. Central canal is going to contain cerebrospinal fluid that was produced in the chambers of the brain. So, big picture now. If your sensation, if your sensory information, you come through the spinal nerve, your cell body is in our dorsal root ganglion, you go through the posterior rootlet, and you enter the spinal cord at the rear at the dorsal gray horn. Then you're going to synapse with another axon, jump into the white matter, and then ascend the spinal cord up to the brain where you can perceive it as a sensation. If we're talking about motor function, you're going to start at the brain. You're going to descend through the white matter. You're going to synapse with the cell body that happens to be sitting here in our anterior gray horn, and then we're going to send an axon out the anterior rootlet. It's going to join up with our spinal nerve, and then that axon will make its way out to whatever muscular organ it's meant to innervate. Now, from a sensory standpoint, we won't really deal with this very much, but the term is going to pop up, called a dermatome. We tend to notice that every nerve root leaving the spine carries sensory information for a very particular part of the skin. Now, this is not going to have a whole lot of practical information for you, but if you've ever had a pinched nerve, you know where that nerve root supplies, because generally you will get some pattern of pain, numbness, or tingliness running along these dermatomes. So, you know, a common one is the L5 spinal nerve. You herniate or pinch the L5 nerve in your back, and you will get pain that goes down the leg, across the anterior part of the shin, and down into the foot. The S1 nerve, however, that pain will radiate down the back of the leg. This one's a little bit more common. You pinch the C6 nerve, and you will get pain that runs down the lateral part of the forearm and into the thumb. So for each spinal nerve, we get a very distinct region of the skin that it innervates. So now we've got all of the information we need now to move on and discuss one of our major topics of the day, spinal reflexes. We are going to look at 3 particularly named and functional reflexes having to do with the anatomy that we just discussed. Now, first of all, though, what is a reflex? We discussed sensory information coming from the periphery of the body, entering the cord, and then going up to the brain where we can perceive it. We talked about motor function starting in the brain, going down the cord, and moving out to the muscle so we can control the muscle. Not all information makes it up to those higher brain centers. Those would be the reflexes. A reflex, by definition, is any motor response that was brought about by a particular sensory stimulus without involving higher brain centers. Sensory in and immediately creating a motor response back out. And once we look at a couple examples here, hopefully that pattern will make more sense. Sensory in, again, remembering our pathway, posterior rootlet into the back of the cord, and immediately synapsing with other neurons to get a motor response back out at the same cord level. Now, generally speaking, what I just drew was called our reflex arc. Now, a reflex arc can generally be 2 or 3 neurons long. The 3 neuron ones are going to be the most common, and those will be the ones we'll be talking about. So they involve a sensory neuron, a motor neuron, and a little short interneuron connecting the two. So to form our complete reflex arc, the sensory neuron detects the stimulus, synapses with an interneuron in the gray matter of the cord. That interneuron synapses with the motor neuron at the same level, and the motor neuron sends a signal out to the muscle. The 3 reflexes we'll be looking at are the tendon reflex, the stretch reflex, and the flexor crossed extensor reflex. As we go, they will become more complex. And as we go, I want you to make note of 3 things about each reflex. This is what you'll have to know about each one. What type of receptor is involved? What the muscular action is? Contraction, relaxation, or some combination thereof. And thirdly, the function of the reflex. Now, all 3 of these reflexes are protective. So when I say function, be specific. What is it protecting against? So those are the 3 things you need to know. The receptor involved, the action at the muscle, and the specific function of the reflex. Before we get started, though, I want to make sure we get some terminology clear. If anybody listening to this right now works in a clinical situation where you take reflexes, you sit on a doctor's table, he gets the little hammer and he hits you on the knee and the leg kicks out. If you are working in an environment where that is being performed, you will hear those referred to as deep tendon reflexes, DTRs. It's worth noting that those are not tendon reflexes. Those actually are a stretch reflex. And the deep tendon reflex is more of a clinical term, but it can be very misleading if you look at how our reflexes are actually categorized physiologically. So starting off, our tendon reflex. First off, the receptor is called the Golgi tendon organ. The Golgi tendon organ sits in the tendon of a muscle, sensing stress and strain. If we were to look at the structure of a Golgi tendon organ, it's going to be encapsulated. And inside that encapsulation, we are going to have these collagen fibers that are all corrugated. They're zigzaggy, they're loose. Picture a bunch of them just packed into that encapsulated receptor. Interspersed amongst those loose, slack collagen fibers are going to be our fine nerve endings that are going to be part of our nerve. They just hang out there in between those slack collagen fibers. Golgi tendon organs sense stress and strain on a tendon. So if we pick up a dumbbell and we start to curl it, stress goes through our bicep tendon and we start to pull on that tendon, stretching our Golgi tendon organs. If we stretch that encapsulated receptor, what's going to happen to those slack collagen fibers that we see in between? It's going to start to straighten them out. When they straighten out, the spaces in between close down and they start to pinch the nerve endings sitting in between, and we will get a signal sent up the nerve giving varying degrees of strain on that tendon. The more we stretch that encapsulated receptor, the more strain is detected. Now, there's limitations to how much the body can take. If we provide enough strain on a tendon, that tendon will rupture. So we want to have a safeguard in place to prevent that from happening, obviously. So what we will see is that if we hit a certain threshold where the amount of strain on a tendon is deemed to be risky, now what we'll do is we'll send that sensation up the spinal cord into the posterior aspect of our cord, synapse with an interneuron. Then that interneuron will synapse with a motor neuron. And that motor neuron is going to be inhibitory for muscle contraction. Remember, we talked about how the nervous system can be excitatory or inhibitory. This is an inhibitory action. That neuron forcefully tells that muscle to stop contracting. So you've just picked up a very heavy dumbbell. You start contracting the bicep muscle, putting strain on the tendon. That sensation ascends the arm into the cord, synapses with the motor neuron. And as a result, contraction of the bicep stops. This is what we see to a certain extent if you witness somebody doing some sort of 1 rep max in a bench press exercise. They lower the weight down, they start pushing it up, and it's moving, and all of a sudden it stops. Part of that fatigue is obviously muscle fatigue, but part of it is also going to be this stretch or this tendon reflex shutting down muscular activity involuntarily in order to protect the muscle and tendon structure. You will see sometimes when this reflex gets overridden. You've heard of people having the drug PCP, where they will have intoxication with this particular drug and they will become somewhat superhuman for a period of time. They might be in a jail cell, strapped to a bed with leather straps, and they somehow break the straps, pull the bed that's bolted to the floor up, and start bending metal. Now, that's going to come as a consequence, because even though you're overriding the reflex, doesn't mean that the tissues that you're having aren't going to get damaged. So now they come down from their high and they have fractured several of their bones. They have pulled several of their tendons away from the skeletal structure. They've ruptured and torn several tendons and muscles. People with seizure disorders will often have a lot of musculoskeletal injuries afterwards because of the intense muscular contraction associated with it that are no longer going to be regulated by this tendon reflex. So it's an inhibitory action at the muscle, forced relaxation, as a result of excessive stimulation to a Golgi tendon receptor. Next up is our stretch reflex. Again, commonly referred to in a clinic as a deep tendon reflex. And if any reflexes are familiar to you, these are the ones. The receptor in this case is called a muscle spindle. Now, I'm not sure how big of a nerd you have to have a favorite receptor, but this happens to be mine. This is a very interesting receptor. A muscle spindle is responsible for detecting stretch, but not just any stretch. It's most sensitive to rapid stretch, real quick stretch. It's also going to be protective for the muscle because quick stretch tends to be more risky for a muscle, causing more tearing. You stretch a muscle nice and slow, it tends to hold up a lot better than a quick jerky movement. But the part that makes the muscle spindle interesting is that it's meant to detect stretch in an organ that's designed to change length. So it's a bit of a conundrum there. So when we look at the structure, forgive this particular diagram, it's not meant to, we're not meant to cover everything on this diagram, but this diagram does give us an idea of how the muscle spindle is structured. Picture we have all these parallel muscle fibers in our muscle organ. In a muscle spindle, we have two contractile portions at either end of the muscle spindle. When you pick up a dumbbell and you start to contract slowly, you get what we call coactivation. The neuron supplying the bulk of those muscle cells for contraction is called the alpha motor neuron. The neuron that innervates the contractile portions of the spindle is the gamma motor neuron. So you pick up a dumbbell and you start contracting it slowly, and both of those types of neurons fire into the muscle, shortening the muscle as a whole with all of our main muscle cells, but also maintaining the overall length of the muscle spindle because we contract each of our poles of our spindle. So even though the muscle as a whole gets shorter, what we've done was maintain the overall length of the muscle spindle. So as you're shortening the muscle and lowering that dumbbell back down, the muscle spindle doesn't detect that as stretch. Because remember, it's going to be really sensitive to quick stretch. Where this spindle springs into action is if there's a bit of a disconnect. If the stretching of the bulk of those muscle fibers happens more quickly than the spindle can keep up with, that's how it senses quick stretch. Knowing that that's the type of stretch that's more risky, that's the sort of stimulus that, again, looking at our reflex arc, I'm going back to an earlier diagram of our arc again. Keep going back to this because the same pattern. The muscle spindles sense that quick stretch, that sensation, ascends whatever spinal nerve it happens to be in, enters the posterior aspect of the cord, synapses with an interneuron, which synapses with a motor neuron, except this time the reflex is excitatory. That same muscle that was getting the stretch now contracts. What better way to counter stretch than with a shortening and a contraction? Protecting the muscle against quick stretch. Now, the two reflexes we've already talked about are what we call ipsilateral, one-sided. But not only are they ipsilateral, but whatever organ had the sensation is the same organ that gets the innervation. So with a tendon reflex, if the bicep tendon is where the receptor is, it's the bicep muscle that is forced into relaxation. For the stretch reflex, if it's the bicep muscle that detects the stretch, it's the bicep muscle that undergoes the contractions. Not only are they ipsilateral, but they're very particular to that same ipsilateral muscle. Now, moving on to our more complex one. Flexor crossed extensor. We see a pretty dramatic pattern of muscle activity here. These reflexes will involve both sides of the body. It's a bilateral reflex. And it will innervate reciprocal muscle groups with alternating opposite actions. Reciprocal muscle groups, for example, bicep versus tricep, hamstrings versus quadriceps. This is a reflex that protects against tissue damage, and the stimulus is what we're going to, at least right now, call a painful stimulus. We're going to refine that term, pain, as we go through the next couple of lectures. The reflex is driven by the receptor called the nociceptor. The nociceptor is what we're going to call, moving forward, a true pain receptor. It detects tissue damage, chemical changes in the tissue that would be an indication of tissue damage. Just like the other reflex arcs, you sense that pain, you send that signal up to the posterior cord, synapses with an interneuron, out the motor neuron, except this time we see patterns of innervation. We also will see this reflex called the withdrawal reflex, because it withdraws the part of the body away from whatever gave you the painful stimulus. You step on the nail, it pulls the foot away from the nail. You touch a hot stove, it pulls the hand away from the hot stove. It withdraws. But when we look at how that is accomplished, it takes on a much more complex pattern than we saw with our first two types of reflexes. First of all, on this diagram, focus on the right side here, please. This is a stretch reflex over here. We already talked about that. So we burn the hand. That sensation enters the posterior part of the cord, reflex arc, interneuron, motor neuron. The motor neuron innervates the bicep to contract, therefore pulling the hand away from the painful stimulus. But here's the rub. That's an excitatory muscle action. But how can we make sure that that hand pulls away from the painful stimulus as quickly as possible? Not only do we want to get a nice quick contraction of our bicep, but we also want to get forced inhibition or forced relaxation from our tricep. So we get reciprocal innervation to those two muscle groups. The bicep and the tricep are antagonistic to one another. If you are trying to contract the bicep, you are having to overcome the stretch of the tricep. So if you want to contract the bicep quickly, the stretching of the tricep becomes a hindrance. What better way to make the bicep move the hand quicker than by, at the same time, forcefully relaxing the tricep? So that part is ipsilateral. That is the withdrawal portion of our withdrawal reflex. But remember the name of the reflex. We call it a flexor crossed extensor reflex. What we just observed on that same side of the body was the flexor component. We flex the arm to pull away from the hand, pull away from the flame. Now, on the other side, it isn't drawn as a bilateral here, because remember, this is a different issue. But that same reflex would also cross the spinal cord. Remember, flexor crossed extensor. There's an extensor component to this reflex happening on the opposite side of the body. So if you want to do a bit of a home exercise here, and you have a spouse that you're living with, a significant other, a roommate, set up like a booby trap, take a hot pan out of the stove and just lay it somewhere where they're going to touch it. I'm joking, of course. You'll be tempted to watch the hand that's about to touch the pan. Don't watch that hand. Watch the other hand. Because as they withdraw from that painful stimulus of touching the pan, the other hand, the other arm will straighten. Flexor on the injured hand, extensor on the opposite side of the body. On the opposite side of the body, they will get contraction of the tricep and relaxation of the bicep. Now, we see this in a more practical situation if that painful stimulus is the foot. You wake up in the middle of the night and you step on a Lego. You will withdraw that foot away from the Lego, but at the same time, the other leg straightens out to support the weight. So you flex the injured one, you extend the opposite. The main reason that that reflex is going to be there for is, again, to give us as rapid a withdrawal as possible. And by doing the opposite reciprocal movement on the other side of the body, it stabilizes the core. So now as the arm is pulling away from the flame in this picture, it's doing so against a more stable anchoring point. By straightening the opposite arm, it prevents more torque of the upper body, and it more quickly withdraws the hand away from the injured stimuli. So nociceptor is our receptor, protective against a painful stimuli causing tissue damage. And the action depends on the muscle, because we see a pattern of excitatory contractile actions and we see a pattern of inhibitory relaxation actions. Finishing up our spinal cord structure is our meninges. It's a orientation of 3 membranes that surround the brain and spinal cord. So this anatomy is going to be relevant to the brain as well. Not only are the membranes important, but the spaces between them actually become just as much of a topic. They're listed here from superficial to deep. And we use the bone as a bit of a reference here. We said vertebra because we're talking about skull now, but this could also, or talking about spinal cord, but this could also be, parietal bone in the skull. And as we move from the bone towards the spinal cord, this is the order of anatomy you would pass through. 3 membranes. On the very surface of the cord or the brain, we have our pia mater, extremely thin and fragile, sitting right on the surface of the neural tissue. The next is our arachnoid matter, still pretty thin and fragile, but not near as fragile as the pia mater. You can get pieces of this off without it just disintegrating. And then superficial to that, we have our Dura Mater. Durable is extremely tough. In lab, if you can get your hands on one of the sheep brains that still has some of the Dura Mater attached, pick it up, yank on it, twist it. It'll feel like a thin leather. Very durable. We have 3 membranes. We have spaces. In between the arachnoid and the pia mater, we have our subarachnoid space, containing cerebrospinal fluid. In the slides beyond this one in your notes, you'll see a description of each space and each meninge. Subarachnoid space contains cerebrospinal fluid. We'll talk about that when we do the brain. Cerebrospinal fluid is produced in ventricles deep in the brain, chambers in the brain. It will leave those chambers through a number of different openings, but it will always wind up right here in our subarachnoid space. In between the dura mater and the arachnoid mater, we have our subdural space containing blood. Now, that's going to be important in relation to our subarachnoid space, because cerebrospinal fluid, you will see some people describe it as a circulation. There is no circulation. It's produced in the chambers of the brain. It makes its way through those chambers, through various openings, winds up in the subarachnoid space. Then its journey is done. It leaves the subarachnoid space by going through the arachnoid mater. Very particular places in the arachnoid mater give a one-way door called arachnoid villi. And that CSF will enter the blood of the subdural space and then be treated like any other waste product. The epidural space, that name might sound familiar because you might have heard of people having epidural shots. with childbirth or pain management. The epidural space, even though we can evoke function from the spinal nerve there to block pain, mostly contains adipose tissue. So you'll have to know the orientation of the membranes and the spaces in between and what we find inside them. And we see them arranged concentrically: Dura Mater, arachnoid matter, and pia mater is going to sit directly on the spinal tissue itself, or brain, depending on if we're in the brain, of course. So it's important to know the structure of a nerve. Please don't get the terms confused with the structure of a muscle. Structure of the spinal cord: white matter, gray matter, the funiculi, the horns, the commissure. We use that anatomy to describe the reflex arc, and then we talked about 3 different reflexes. You need to know the receptor, you need to know the function, you need to know the action of the muscle, because those are all musculoskeletal reflexes. And finally, the arrangement of the meninges that surround the spinal cord, and of course that arrangement and spaces are going to be exactly the same if we were talking about the brain. So from here, again, we're moving our way into the central nervous system now. We just did the spinal cord. We're going to talk about the superior structures now of the brainstem and the brain next. And if you have any questions, please reach out and let me know. Thank you.